Okay, welcome to this lecture video. My name is Ferdy Hellweger, and we're going to be talking about modeling antibiotic resistance in the ambient aquatic environment. Before we get started, let's clarify about the intended audience and the objectives of this lecture. So the intended audience is students in graduate level surface water quality modeling courses. I teach surface water quality modeling here at Northeastern. It's a graduate course elective in our environmental engineering program, and I cover it towards the end of the course as an advanced topic. Another audience is researchers interested in using the McKee model. This is the model that I'm going to be presenting for their research projects. So the objectives is to provide an introduction to antibiotic resistance and water quality modeling, as well as the McKee model, and then provide a hands-on tutorial for the McKee model. So in terms of outline, I'm going to be starting with a relatively broad introduction, antibiotics and antibiotic resistance, antibiotic resistance in the aquatic environment, water quality modeling, and then how antibiotic resistance relates to water quality modeling, how those two separate fields merge, or how water quality modeling can be applied in the context of antibiotic resistance. Then I'm going to be giving a description of the McKee model. I'm going to present an application of the model to the Poudre River. And then we're actually going to leave this slideshow, and I'm going to give you a hands-on demo where we download the model, specify some input, run the model, interpret the output, and then I'm going to be end with a homework assignment. So let's get started with the introduction. Antibiotics. So probably one of the most significant tools in our fight against diseases are antibiotics. They've done wonders in extending the life expectancy. One of the earliest antibiotics was penicillin, and here is a picture that shows the significance of it. A lot of people used to die due to infections on the battlefield, and really penicillin has changed that quite a bit. Of course, in this case, it's a bit ironic. It would have been easier not to go to war in the first place, but there are a lot of infections that are also unrelated to us fighting and killing each other. So penicillin was the first antibiotic. Now there are many, and probably on the order of 100 or so, and they vary in their mechanism of action, how they affect the cell. And this slide shows some major targets of common antimicrobial agents, and you can see that they can affect various functions of the cell, including the cell wall, either breaking up the cell wall or preventing the synthesis of the cell wall. They can interfere with the DNA replication or with the synthesis of proteins by the ribosomes. That is also the mechanism of action of tetracyclines, which is what we're going to be focusing on a lot in this lecture. So tetracyclines bind to the ribosomes and prevent them from making proteins. Okay, and if a cell cannot make proteins, then it cannot grow. And so tetracyclines prevent the growth of bacteria. So unfortunately, bacteria relatively rapidly develop resistance to antibiotics. And here is an example. This shows the percentage of resistant strains found in blood culture. In this case, methicillin resistant staphylococcus. And you can see that in a period of about 10 years or so, the resistance has gone up consistently, relatively rapidly. And we're now at a point where there are many antibiotics that are simply ineffective. There are many bacteria, we refer to them as superbugs, that are resistant to all of the antibiotics that we have in our arsenal. And so you can see it's a major public health concern. Antibiotic resistance. Let's talk a little bit about the mechanism of antibiotic resistance. Here's a simple cartoon that illustrates some ways in which a cell can become resistant to an antibiotic. One common mechanism is efflux pump. So that's an enzyme on the cell membrane that basically takes the antibiotic and pumps it back out, keeping the concentration of the antibiotic relatively low inside the cell so that it can't do any harm. There are also enzymes that alter the antibiotic or simply degrade or destroy it so that it cannot function. This is not an exhaustive list of resistance mechanisms. There are others, including, for example, altering the site of action of the antibiotic. And so the resistance mechanism is commonly encoded, or quite often encoded, on plasmids. Those are extracellular extrachromosomal, not extracellular, extrachromosomal DNA elements. And this is significant because what can happen is these plasmid can be exchanged amongst bacteria. Okay, and so there are various mechanisms of transferring the resistance from one bacteria to the other, not only vertically from the mother to the daughter cell, but also laterally or horizontally to other cells of the same strain species, or also other species. Okay, and there are three mechanisms that are responsible, or that can do this. Genes can be transferred simply by taking up DNA from the environment. So if a cell breaks up, then another cell may take up some of that DNA, and that may include the resistance gene. There can be conjugation, where two cells actually touch and transfer DNA from one cell to the other. And then the transfer can also be mediated by viruses. Viruses can transfer and bring DNA from one cell to the other. So that's sort of a brief overview of antibiotics and antibiotic resistance. And we are now starting to see some of these antibiotics and antibiotic resistance in the ambient environment. Here is an example. There are many papers out there that are now documenting that when we go out into the environment, rivers, lakes, etc., we look at how many of the bacteria there are actually resistant to antibiotics, we find that the number is relatively high, especially in the anthropogenically impacted environment. So this shows an example of one study where you see a river here on the left and shows the sampling locations, and what's shown here is the percentage of resistance strains to various antibiotics. So TE, for example, is tetracycline, and you can see that Aeromonas has about 20% of the strains are resistant to tetracycline. And if you look at the total, you may get up to 70 to 80% resistance. So this is an obvious concern, because we're concerned that this resistance may end up being passed on to pathogenic bacteria that then impact us and may contribute to making our pathogenic bacteria that cause diseases more resistant. So we're very concerned about this problem. So let me sort of switch tacks a little bit and talk about water quality modeling, and then we're going to see how that can be related to antibiotic resistance. So water quality models have been around for a long time in environmental engineering, and they are very well established and accepted as useful tools. for management as well as understanding of environmental systems. They can be used in the traditional manner in environmental engineering to relate discharges to ambient conditions. And so you need that information to design and place a wastewater treatment plant. They also can be used for interpreting field observations and hypothesis testing. What I'm showing you here is an example of a eutrophication model that was developed in the 1980s for the San Joaquin estuary. And you can see that what we commonly do is we divide a water body up into mass balance segments, and then within each segment we define various state variables that tend to be concentrations of compounds like nutrients. We run the model and we get a prediction, and we compare the model to observations to confirm that it is an adequate representation of the system. And then we may use the model to evaluate management scenarios, such as reducing the amount of nutrient inputs. So, you know, water quality models have been around for a long time in environmental engineering, and they are continuously evolving to address changing societal problems. You know, one of the earliest problems that environmental engineers were faced with was the depletion of dissolved oxygen in a river due to the discharge, a point source discharge of raw wastewater, and this prompted the development of the Streeter-Phelps model that you probably would be familiar with if you've taken environmental engineering undergraduate courses, also commonly referred to as the dissolved oxygen sag curve model, and that, you know, maybe or is often considered sort of to be the first water quality model. But then, as we were faced with other problems, pathogens, eutrophications, toxics, and so on, we continued to develop mathematical models to help us manage and understand these problems. At the forefront, the problems that are new to us these days are pharmaceuticals and personal care products, and antibiotics themselves may be considered to fall in this category. And then maybe the most cutting-edge problem is antibiotic resistance. So it only makes sense for us to start developing water quality models for antibiotic resistance if you look at the history of environmental problems and the role of water quality modeling. Now, it's a bit of an uncharted territory. You know, oftentimes when we deal with a new problem, the mechanisms are not so different from what we've had in the past. Oftentimes, many of these problems that our models addressed have mechanisms like sorption, decay, speciation, growth, grazing, and predation. When we're dealing with antibiotic resistance, we're faced with more or different mechanisms, and that's a bit challenging. So we have to consider things like the effect of the antibiotic and metals on the bacteria, the cost of carrying the resistance, the metabolic cost associated with carrying the resistance plasmid, for example, the transfer of resistance as well as the loss of resistance. All those are a bit uncharted territories to us. Okay, so hopefully I've made the case here that antibiotic resistance and antibiotic resistance in the environment is an important problem, and that water quality modeling is a tool that may help us manage and understand this problem in general. So let's start talking a little bit more specifically. What I want to talk about in the context of this problem is a question. I've mentioned that we see relatively high concentrations of antibiotic resistance in the aquatic environment, and one question that may come up is why are they there? And there are basically three scenarios or hypotheses. One is that there's relatively large input wastewater treatment plants or agricultural discharges have, because of their the nature of what they do, have relatively high numbers of antibiotic resistant bacteria. And maybe the reason why we see these high levels in the environment is simply because they're being discharged at these large rates, but they don't really grow to an appreciable amount in the ambient environment. Another scenario is that the input is relatively small, but the resistant bacteria in the environment grow because the wastewater discharge also includes antibiotics. Okay, so antibiotic resistant bacteria are being discharged along with the antibiotics, and the antibiotics holds down the susceptible bacteria, and that allows the resistant bacteria to grow in the vicinity of these discharges. Another scenario, scenario H3, is again that the input is relatively small, but in this case it's not the antibiotics that is holding down the susceptible bacteria and promoting the resistant bacteria, but it's metals. Okay, and this is a mechanism referred to as co-selection, and the idea here is that oftentimes what happens is the same mechanism that makes a bacteria resistant to an antibiotic also makes it resistant to a metal. Or the gene that is responsible for the antibiotic resistance is on the same plasmid as the gene that's responsible for the metal selection. So if you select a bacteria that is metal resistant, it is more likely to also be antibiotic resistant. Okay, so let's talk about the model. So we want to explore these hypotheses, and let's talk about the model. This is what we refer to as the kinetic diagram. Okay, so this is very similar in concept to the kinetic diagram that you saw for the eutrophication model that I introduced in the introduction. Each one of these gray nodes here, or boxes, is a state variable, and we have a segmentation that covers the water column and the sediment bed. So what we have is we have susceptible bacteria, XS, and resistant bacteria. These bacteria both grow on dissolved organic matter, and the growth of the susceptible bacteria is inhibited by antibiotics as well as metals. The resistant bacteria is not inhibited because it's resistant. Okay, there can be flux from resistance to susceptible and back and forth, and this is that exchange of resistance that I talked about in the introduction. So Kc is the mechanism that refers to conjugation, so susceptible bacteria becomes resistant, and Ks is what we refer to as segregation. That's when a resistant bacteria loses the resistance plasmid. The antibiotics and the metals partition to dissolved organic matter and particulate organic matter or total suspended solid, so there's sorption involved, and the particulate organic matter becomes dissolved organic matter. Okay, and so that's an overview of the mechanisms. Then there is transport in the water column and also to and from the sediment bed. And then we have the same types of reactions going on in the sediment bed. Let me cover some of the equations here. A few slides with equations. This is a general mass balance for transport in the water column and the sediment bed. What it says is that the change in concentration in the water column is a function of the settling. Mass is removed by settling. The particulate fraction of the chemical is removed by settling. There is a gain by resuspension from the sediment. there may be diffusion to and from the sediment. Then there is also horizontal transport via inflow and outflow, as well as external sources such as wastewater discharges, which follow the common water quality modeling approach, and I'm not presenting those here. In the sediment bed, you may have settling as a source from the water column. You have resuspension as a sink or a loss, and again, there is diffusion. If you get into the details of these equations, sometimes there are some nuances. For example, the reason why I have C prime here and not C is because we have the option of expressing the concentration either on a pore water basis in the sediment or on a total volume basis. That is all described in common water quality modeling books, and all those things are correctly accounted for in this model. So antibiotics and metals, relatively simple approach is being used for modeling those. The antibiotic decays in a first order manner. Again, now in these mass balance equations here, I'm omitting the transport that I've outlined in the previous slide. So antibiotics decay in a first order manner, and metals are conservative. One mechanism that's important is partitioning. So we use linear partitioning, and so we may calculate the freely dissolved fraction of the antibiotics by partitioning to dissolved organic matter and solids. And in a similar manner, we may calculate the freely dissolved metal concentration. This approach is the same as what is outlined in common water quality modeling textbooks. It's the same approach we use for example, simulating PCB transport. So we can calculate the freely dissolved fraction or freely dissolved concentration. That's going to be the concentration of antibiotics and metals that affects the growth of the bacteria, the susceptible bacteria. We can also calculate the dissolved concentration, which includes the freely dissolved as well as the DOM bound, and that's what's going to be important for diffusion to and from the sediment bed. And then again, when you're dealing with the sediment bed, you may have to account for the porosity of the sediment bed, and again, that's all accounted for correctly in the model. But I'm not covering those details here. Okay, so the bacteria. Again, I start with the mass balance equations. The susceptible bacteria change as a function of growth, so they grow, and their biomass reduces as a function of respiration. And then again, we have transport, which is omitted here. The same equation applies to the resistant bacteria. And let me outline the growth rate. Okay, and this is really where this model is new, or where there is new concepts introduced in this model. So the growth rate of the susceptible bacteria is a function of the maximum growth rate, and then a Michaelis-Menten or Monod type limitation term. So the more dissolved organic matter, the higher the growth rate. So this is limitation by the nutrient or the substrate. And then the growth rate is reduced due to the action of antibiotics as well as metals. Okay, and what's shown here is an equation that essentially works out to be a linear effect of the toxicant. Okay, so the growth rate reduces in a linear manner with increases in antibiotics or metals, and that is based on that is true for tetracyclines, which is what this model was developed for, as well as the metals that we're considering, and experimental evidence support this, and you can follow the references here on the bottom if you want to learn more about this. So the toxicity of the antibiotic is quantified using the MIC, which is the minimum inhibitory concentration. When the concentration is at the MIC, then their bacteria are not growing. For metals, we quantify the toxicity using an IC50. When the concentration is at the IC50, the growth rate is at 50%. Okay, but again, the effect is linear, so essentially the MIC is equivalent to 2 times the IC50. So that's the growth rate for the susceptible bacteria. The resistant bacteria has a similar equation to begin with. There is a maximum growth rate, and then there's limitation by dissolved organic matter, but there's no effect from the antibiotics and the metals, right? They are not affected because they're resistant. However, the resistant comes at a cost, right? So there is a metabolic cost associated with carrying the plasmid, with expressing the efflux pump and things like that. So as a result, the resistant bacteria growth rate is reduced compared to the susceptible one. And that is quantified using this alpha term, the cost of resistance. So that's an overview of the model. Now we're going to get into the application. We've applied this model to the Poudre River in Colorado. The Poudre River is a test bed for studying antibiotics and antibiotic resistance in the ambient aquatic environment. A relatively large database has been developed and continues to be developed in the research groups of Ken Carlson at Colorado State, as well as Amy Pruden now at Virginia Tech. And so it's an ideal test bed because it covers pristine area in the upstream reaches, and then there are 2 urban centers and relatively large agricultural area. So it's a relatively diverse setting, and so this is an ideal application for our model. So we applied the model to this river, and what I'm showing you here is the result. I'm showing in the top panel the toxicants. So this is tetracyclines, as well as 3 selected metals, copper, zinc, and selenium. The concentration versus distance along the river. And I'm indicating here the approximate location of Fort Collins and Greeley. Those are those 2 urban centers. Okay, and so this is relatively established water quality modeling, right? Modeling of toxicants and metals. And you can see by comparing model to data that there are some discrepancies between the model and the data, but the model does capture the main patterns of what we see. Specifically, an increase in concentration of toxicants at Fort Collins, and then maybe for the tetracyclines another increase again at Greeley. Okay. On the bottom panel, I'm showing you the resistance. All right, so this is the resistance, the density of resistant bacteria in the sediment bed, and you can see that it's relatively low, and then there is an increase, a jump up at Fort Collins, a decrease, and an increase again at Greeley. Okay, so we now want to use the model to simulate this resistance, and what I'm going to do is I'm going to go through those 3 scenarios that I introduced earlier, the 3 hypotheses. If you recall, the first hypothesis was that the reason why we see these high levels of resistant bacteria is because of a large input from wastewater and other sources. So to explore this hypothesis, we run this model by setting the amount of resistant bacteria in the effluent of wastewater treatment plants very high, to the maximum that has ever been observed anywhere. And also we make them all particulate, so they sink right away to the sediment bed and are not washed out. So this is a relatively conservative scenario designed to maximize, to see how much can be explained, how much of the resistance in the sediment can be explained by these mechanisms. And the results show that we cannot explain the observed resistance. Even if we maximize out the resistance in the effluents and we make them all particulates, so they send to the sediment bed, then that does not explain the amount of resistance that we see in the sediment bed. So this is an important conclusion. The resistance cannot be explained by external input alone, but there has to be some growth. Okay, so the second scenario, hypothesis H2, was that it's the antibiotic that results in their growth and over the susceptible bacteria. So we run this model, we dial back the input of resistant bacteria from wastewater treatment plants back to more reasonable levels, as well as how much of them are particulate to more reasonable levels, and we include the toxicity of the antibiotic, and there we see that we basically do not see any resistance. Okay, the concentration of antibiotics that we see in the ambient environment are many orders of magnitude below what we know to be toxic for Tetra2 bacteria. So really those low levels of tetracyclines don't exert any significant advantage to the resistant bacteria. The third hypothesis is co-selection by metals, and we run three different scenarios: copper, zinc, and selenium, and these are the results shown here. And what we see is that with the copper and the zinc scenarios, we get resistance of the same order of magnitude, or at the same level as what we observe. The selenium, we see some resistance, but not at the level of what we observe. Now, there is some calibration involved here, but the point is that we can calibrate the model to match the data within the observed or literature range of the parameters. We were not able to do that for scenarios H1 and H2. Okay, even if I max out the input from the wastewater treatment plants, I cannot explain, I cannot make the model reproduce the observed resistance. However, with copper and zinc toxicity, I can calibrate the model so that we observe, it predicts levels of resistance consistent with what we observe. And the pattern, the general pattern, is somewhat consistent as well. We have an increase at Fort Collins, a decrease in between, and then another increase at Greeley. Here's just another picture of this. This shows the zinc co-selection case, a GIS map of model and data. So the model river segments here are colored and sized based on the model predicted resistance, and the data are also shown here with the symbols. So the main conclusions that we reach are that the density of antibiotic resistant bacteria cannot be explained by input from sources or selection by antibiotics, but it can be explained by co-selection by metals, specifically copper and zinc, but not selenium. There is significant parameter uncertainty, something that I have not talked about a lot in this lecture, and I'll refer you to some of the publications that I'm going to list later on for the details. But probably the most significant source of uncertainty is the cost of resistance. We don't know what it costs a bacteria to be resistant in the ambient environment. There are a lot of data for hospital or clinical strains, but not for ambient strains, and that constitutes a significant source of uncertainty. Okay, so now let's go to the demo. We're going to go through a hands-on application of the model, and I'll just highlight to you you basically need 3 files, and we're going to go through the download here together: model, run x1, and figures. And run x1 is a model scenario that includes copper and tetracyclines together. You can download the files from my website, or you can use this direct link. So let's leave the slideshow, minimize this, and open our web browser. We're going to actually download from my homepage the model, so www.systemsbioecology. If you go to models and code, you will see that there is a Maquis model, so we're going to download that. We're going to click on that. We're going to download that, and let's just save that on the C drive in a temporary directory. I'm going to save that. We can open the folder. Let's close this WinZip application. You should be able to just open it using your Windows Explorer. Let's try this. So the Maquis model has a number of files here. I'm just going to take these and I'm going to drag them into the temp directory, and that'll basically unzip them. Okay, and so now I will use these directly here. Okay, so the files that we have that are important are figures, model, and run x1, and I'm going to open those. Okay, now there are 3 files: figures, model, and run x1. The reason I could put the model in just one Excel file. That wouldn't be a problem. But what I like to do often is I like to have multiple scenarios. I may run a copper scenario and a tetracycline scenario, etc. And then I may want to have multiple of these runs. Okay, but there's no reason to reproduce the code which is in the model for each one of these runs. Okay, so the model sheet, all it has is the code. The run x1 in this case has the input and the output. And then the figures is just a figure that references the run. And again, I keep that separate from the runs because I may want to compare multiple runs on multiple runs in one sheet. Okay, so let's take a look at the figure first. So what we have here is simply just one figure that compares the tetracycline and copper concentration as well as the resistance. This is very similar to what you saw in the presentation. The model sheet has the code. When you open it, you won't see anything. That's because the code is in the worksheet as Visual Basic code. Okay. Now this may cause a problem when you open the model first on your computer, because oftentimes due to security, your Excel settings may prevent running of macros. If that's the case, what you have to do is you have to go to File, Options, Trust Center Settings, macro settings and make sure that you've enabled all macros. Okay, that is required to run the model, and in my case that's already the way the settings were. If you want to look at the code, you don't have to. You can run the model directly without looking at the code, but if you want to look at the code, you can go to View, Macros, View Macros, I'm sorry, let's try Enable Editing. You may go to View Macros, View Macros, and there are macros, and if you just go to Edit, you can see the Visual Basic code here. So this can be modified or looked at if you are a developer. I'll just make one note about the use of Visual Basic for this model. It's very easy to use Visual Basic because you can read and write directly to and from the Excel sheets. The disadvantage of using Visual Basic is that it's relatively slow compared to other programming languages such as Fortran. That's not necessarily a problem depending on your application, but it may be. And actually it turns out that in our most recent project we did a Monte Carlo analysis, a parameter sensitivity analysis, where we had to run thousands of simulations. So I actually recoded this in Fortran, and if you're interested in or if you find that the runtime is an issue, you can contact me and I will gladly provide the Fortran code to you as well. Okay, so let's go at the look at the run X here. This is the model input and output, and again I'm going to enable editing here. So this is the model input and output. The model reads directly from the Excel cells and it writes to the Excel cells. Okay. The input is in these green sheets here. Okay. There are constant inputs, spatially variable inputs, and time variable inputs. There are a couple of figures here that plot the results. There's a spatial plot. These are all the state variables, how they change with distance. And here's a time variable progression, how all the state variables change with time during the model simulation. And then there are outputs. Let's start with the input. Okay, so what we have is, I'm basically going to go through all of the inputs here. And it's divided, so the constant inputs are divided into sections. There are, the first section is run control. So that specifies the time step used by the model, the end time. And what you see here is oftentimes I have cells shown as red and green. Green cells are designed for you to type into, and red cells are calculated. So here I found it more convenient to specify the time in years, but the model takes the time as days, and so it calculates the end time as a function of what you type in here. It's relatively straightforward. We also specify a print time step and a print start time when you would like to start printing. You can specify how many chemicals, number of chemicals and number of bacteria you want to include in your simulation. In this simulation, it's actually 2 chemicals, copper and tetracyclines, and 1 species of bacteria. You specify for the segmentation, the dx, the spatial segmentation. Each segment is 1,000 meters or 1 kilometer long, the number of segments, and the depth of the sediment layer. Hydraulic geometry parameters specify the width, the depth, and the velocity for the water column. You can specify a dispersion coefficient for the water column. In this case, we specify 0. That is because all of the dispersion, or the amount of dispersion that is introduced by numerical dispersion, is sufficient, or approximates the actual dispersion in the system. So we don't actually need to add any additional dispersion. Sediment transport. We have a settling velocity for solids. We specify what fraction of the bacteria are particulate. We have a resuspension velocity and then a diffusion velocity to and from the sediment bed. Then we specify initial conditions. And what you see here is there is actually, the model is set up to simulate 10 different types of bacteria. So you can specify the concentration of susceptible bacteria for bacteria 1, 2, 3, etc., all the way to 10. In this case, we only use one bacteria type, and therefore we only specify an initial concentration for one bacteria. When there are two cells showing up, it's typically the initial condition for the water column and then the sediment bed. Okay, so the water column concentration is 0.02 milligrams per liter and around 500 for the sediment bed. Initially, there are no resistant bacteria in the sediment bed, right? So the initial condition for resistant bacteria is 0. You also need to specify initial conditions for particulate organic matter and dissolved organic matter, as well as for the chemicals. And for the chemicals, it's all 0. We start with a clean river, if you want to look at it that way. We specify the boundary conditions, again, for our 10 different bacteria and our chemicals. Okay, and let's just jump right down to the chemicals. We specify different conditions here for pristine, confined animal feeding operations, other agricultural, urban, and wastewater. So we specify different concentrations for these types of land uses or point source inputs. Okay, and you'll see how that is being used spatially on the next sheet. Okay, so for example, the copper is chemical number 1. The wastewater treatment plant concentration is 0.011 milligrams per liter or 11 micrograms per liter. Different concentrations are specified for the different land uses, and the same thing for tetracyclines. In terms of kinetics, we have an option in the model to decay the bacteria, which was not used in this application. So you can specify a decay rate for the susceptible and the resistant bacteria, the particulate organic matter, as well as the chemicals. Copper is chemical number 1 and it doesn't decay, but tetracycline decays at a rate constant of 1 per day. We have partitioning of the antibiotic or the toxicant. In this case, actually, it's chemical 1 is the copper. This is the partition coefficient of copper to the solids, and this is that for tetracyclines, and these are the partition coefficient for dissolved organic matter. We specify for the bacteria the maximum growth rate in the sediment bed, in the water column and the sediment bed. We specify the half saturation constant. We specify the yield, how much bacteria are being produced for a certain dissolved organic matter quantity consumed. And we specify a respiration rate. We also specify the effect of the toxicant. Okay, again, the toxicant for toxicant 1 is copper. We specify the toxicant minimum inhibitory concentration here, and also here for the antibiotic tetracyclines. There is segregation and conjugation. This is the gain and loss of resistance or transfer of resistance between the susceptible and the resistant bacteria, which is not used in this application, so those rates are 0. Alpha is the cost of resistance parameter, so in this case there's a 4% cost of resistance. And then there are a number of other parameters related to the solids. We specify the solids in the water column and the sediment bed, as well as the porosity in the sediment bed, and hydrolysis and the POM and DOM production rates. Okay, so those are all the constant parameters. Here is the sheet for the spatial parameters. Okay, what we have here is each column corresponds to one water column segment, one of the segments in the model, right? And so they are numbered with I, and they have a kilometer distance associated with them, as well as a meter distance associated with them. For each segment, we specify the flow rate, and in this application, it's a relatively complicated setting, because what happens is we have withdrawals from the due to agricultural withdrawals. So what actually happens in this river is that the flow rate decreases as you go along the river, and then it increases again as the agricultural flows are returned to the river. So what we have here is for each segment is the inflow, upstream inflow, the wastewater discharge, the non-point source discharge. This is the runoff unaltered. Okay, this is what you would get from runoff, and then the difference, which is essentially the agricultural or other or municipal withdrawals of water from the river. And then the model does a flow balance here to balance the water. Then we specify the point source concentrations. Again, we have our bacteria, susceptible and resistant. We have dissolved and particulate organic matter, and we have our chemical concentrations here. So let's take a look. Concentration 1 is copper, and our concentration is 11 micrograms per liter. And this just simply references the constants sheet that we talked about previously. Now for tetracyclines, it's a little bit different, because what happens is we know the dissolved concentration, but the model takes into account, or the way you specify input to the model is it's a total concentration. So actually for tetracyclines, we specify a dissolved concentration here, and then it calculates the total concentration based on a simple partitioning calculation. Okay, so it looks a little bit intimidating when you first look at it, but it's relatively simple. So there's an option of specifying the dissolved concentrations here, and then there's a flow rate. And so let's just take a look here. For most river segments, the flow rate is going to be 0. But over here, we're actually at the discharge of the Mulberry wastewater treatment plant, and there we have a discharge of 0.11 cubic meters per second. Okay. Something similar happens with the non-point sources. We have concentrations of bacteria and susceptible and resistance, dissolved organic matter, particulate organic matter, and then concentration of copper and tetracyclines. It gets a little bit difficult again for this specific application, because we assign different concentrations to different land use types. And so actually, if you go down to the bottom here, what you'll see is that there is calculated from GIS the fraction of agricultural, the fraction of confined animal feeding, and the fraction of urban land use draining to this specific segment. Those fractions are used along with the concentrations specified in the constant sheet to calculate the fraction or the concentration that's actually coming in with the non-point sources in this specific segment. Okay, so it looks a little bit complicated because the hydraulics and the way the setting of the Poudre is pretty complicated. You can simplify this quite a bit if you're modeling a river that's much simpler hydraulically or hydrologically. Okay, so here's the again the flow of the inputs. We also calculate some of the geometry using the coefficients that we specify in the constants. Here's an estimate of the numerical dispersion and other miscellaneous things that I'm not going to cover in detail. So that's the spatial input, a brief overview of it. There's also an option for specifying time variable input, and this illustrates it here, but it turns out that actually we're not using any time variable input in this application. It's all steady state. But if you wanted to change the concentration of point source or non-point sources in a time variable manner, you may specify a time series here, so you can simulate fluctuating input. Okay, so that's all the input. What I'm going to do now is I'm going to run the model, and then we're going to look at the output. And so before I do that, however, what I want to do is I actually want to make a change in the model. Okay, so we're actually going to go and back to the constant sheet. We're going to look at the concentrations here, and we're going to change the wastewater concentration for copper. We're going to simulate a reduction in the concentration of copper and the wastewater influent, and we're going to basically simulate a 30% reduction. So I'm going to just multiply this concentration by 0.7. I'm going to reduce the inflow concentration of copper. And let's just, before we start the model, take another look at the figures. This is what the model looks like. Enable editing. This is what the model looks like before we made this change. And now I'm going to run it. And I'm going to go back to the X1 sheet. And to run the model, I'm just going to hit Control M. And what you'll see is it'll disappear. And on my computer, it takes about 25 minutes to run. It may be different for your computer. So I'm going to actually stop the recording here, and then we're going to come back when the model has finished. Okay, so we are back. The model has finished. Let's take a look at the output. First, we're going to take a look at the figures, and if you remember what we were just looking at in terms of this plot, you'll realize that the copper concentration decreased a little bit, but the resistance actually decreased significantly. And that is simply due to the nonlinear relationship between the concentration of copper and the concentration of resistant bacteria. The one thing that I still want to cover while we have these Excel sheets open is the output in the model. Those are the blue sheets here, and I'm just going to want to run down so you're familiar with what's in those. So these are all sheets marked with out. The state variable, so out XS is the output for the susceptible bacteria in layer 1, or the water column. There is also out XS2, those are the susceptible bacteria in the sediment bed. Okay, so let's take a look here. It's structured in the same way as the spatial input. What we have is each column here represents one segment. And then there is output for different print times. Okay, so we're printing a number of times here, and these are the times in years, and you can see how the concentration changes. For the susceptible bacteria, because the model can actually simulate 10 different types, you'll see this block repeated further down several times. Okay, so the first block here is for the first bacteria type. The second block is for the second bacteria type, which in this case we're not modeling or we're not using, so that's all going to be zeros. Similar setup for the resistant bacteria. Sometimes I added calculations down here. If you want to get the ratio of resistant bacteria, they can be calculated as such. Particulate organic matter, dissolved organic matter, dissolved organic matter in the water column, and you'll see there's a difference here. These are both in the water column, DOM1 and DOMW1, but W1 is on a volume water basis. Okay, so you can quantify the concentration on a volume total or volume water basis. For the water column, they're going to be the same, but for the sediment bed, because there's porosity and pore water is only a fraction of the total volume, those are going to be different. Okay, total organic matter, the concentrations of chemicals. Okay, again, what we have here is the concentration of chemical 1, that's the copper, and then the concentration of chemical 2, which is the tetracyclines, and then the rest are 0. For the chemical, we're also outputting the truly dissolved on a water basis. Out C chemical truly dissolved on a water volume basis in the water column, and the dissolved on a water volume basis in the water column, and the particulate one. Okay, and then the same thing is repeated for the sediment bed. Okay, that's basically all I want to cover here for the Excel sheets. Let's get back to the slideshow. Let's start up the slideshow again. I just want to finish with a simple homework assignment. The assignment consists of reading the most recent paper. It's actually included, the PDF version is included with the distribution of the McKee model. Writing a short review of the paper. Then your task is to download the Poudre River model and evaluate a management alternative. For example, or as a first step, you may decrease the copper concentration in the urban runoff by a factor of 2. Similar to how we've decreased the wastewater treatment plant effluent by 30%. And you may try some other management actions that are designed to reduce the antibiotic resistance in the river. You will discuss your results, and then I think it'll be interesting if you try something else. Use your imagination. Make some change in the model to understand what's happening to the antibiotic resistance in this river, and then discuss those results. In total, your write-up should be 1 page of single-space text plus any figures that help you document your simulation. And this gets us to the end. If you're interested, here's some further reading on the McKee model. There are basically 3 papers that document it. And with that, I hope you've learned something and enjoyed this lecture video. Okay, thank you. Goodbye.